All right, let's talk modes of action of antimicrobial drugs. So here in figure 14.9, they kind of draw this out. So let's work from the outside of the cell in. First, the cell wall. This is the most common mode of action. This is a huge group of antibiotics. And this is a good target because all bacteria have a cell wall and our own cells do not. So these do more harm to bacteria. They have better selective toxicity. They do less harm to host cells because our cells don't even have that part. So you'll notice here the penicillins, anything that ends in cillin, like amoxicillin or ampicillin, they're all in this group. They all work by interfering with or messing up or damaging the cell wall. Moving in, the plasma membrane. Another mode of action is by damaging the plasma membrane. This one is less common because our cells have a plasma membrane too, so it's harder to find ones with selective toxicity that damage the bacteria plasma membrane without damaging ours. Then if we move inside the cell, this next one, the ribosomes. So ribosomes in prokaryotes are actually a different size and shape and kind than the one in eukaryotes. So this is the other big one. This is major. This is another huge group of antibiotics that target the bacterial ribosomes. I'll talk more about that in another slide. There's also a small group that targets metabolic pathways where they interfere with the enzymes or block the chemical reactions or compete with the normal substrate at the enzyme and just generally get in the way. So the bacteria can't build or make or use the molecules it would usually use, and it kills off the bacteria that way. These are less common. There's a few of these, but not as many as ribosomes and cell wall. And then finally, down in the middle of the cell in this nucleoid region, the fifth mode of action is to get in the way of DNA or RNA synthesis. So that's the process of copying and making new DNA or RNA. This is another one where there's a few, but not as many because our cells have DNA as well. So it's hard to achieve that selective toxicity where you're interfering with or harming the bacteria cell. It has to be really specific to the proteins and enzymes they use without messing up the proteins or enzymes that we use with our DNA. So the big two groups that most antibiotics are messing with the cell wall and messing with the ribosomes. Smaller groups for these other three, and those are the 5 major modes of action.